This will be a relatively quick overview of a typical EEG and MEG analysis pipeline in M&E Python. Can't cover everything, of course, but I think it'll cover the most important processing stages of EEG, MEG analysis in a nutshell, and we will then unpack them a bit more in the following sessions. And I'm assuming that most of you are familiar, at least, with the basic approach that we take in cognitive neuroscience and functional imaging, neuroimaging. So how we do experiments and what we are trying to achieve, but still I want to hopefully take everyone with me. So I'll start at the beginning. One slide here illustrating where these data actually come from. So they are from the M&E Python sample data set, which is provided with the software. And here one participant was presented with relatively simple stimuli. So it's not exactly an experiment that gets the cognitive neuroscientist very excited. But for illustration purposes, I think it's great. So one participant was presented these simple stimuli like checkerboard patterns to the left, visual field, visual left, same to the right, visual right, also simple tones to the left ear or to the right ear. Interstimulus interval was 750 milliseconds, so every stimulus was presented individually, one at a time. And of course, they were presented in randomized order dozens or hundreds of times, I think for about 4 minutes. And then they recorded EEG and MEG signals. So you may only be interested in EEG, others may only be interested in MEG, but they recorded both. This is just a subset of channels. And then you get these continuous EEG and MEG signals that just like that don't really tell you anything. So they look more or less the same for every participant. And somewhere hidden in these data are the responses evoked by the individual stimuli. And in order to do something meaningful with the data, we need to know when our stimuli were presented, so we can then analyze the particular latency ranges and combine the data across trials. So we do that by sending triggers, which are here just marked by colored lines in the data. So the onset of every stimulus is marked usually by sending a number to the acquisition machine. So it could be number 1 for one stimulus, number 2 for another stimulus, and so on and so forth. So that's up to the experimenter. And then we can cut these data segments out around these stimuli and average them or do something more clever with it. But in the following, we will simply average separately for the different stimulus categories. Then we get something like that. So these are already averaged responses, I think over about 70 or 80 trials. This is just for illustration purposes. So there is a baseline period before the stimulus is presented. We just see the brain noise here. Then there's an evoked response. It looks slightly different for the different stimulus types, but the idea is the same. You cut these data segments or epochs out of your data and you average them together, and then you get something like that. What else is there to say here? I think that's okay. So then you might be interested in a particular latency range or peak in these data. So you can look at topographies, for example. So in this case, I'm presenting EEG topographies. For the stimulus, visual stimulus to the left, you see something here on the right, in the back of the head. So contralaterals to stimulus presentation just before 100 milliseconds. At 90 milliseconds here, you might not be able to read that. So that makes sense. Similarly, for the auditory left stimulus, you get a nice pattern, dipolar pattern here on the right. Visual stimulus to the right, contralateral response, posterior left, scalp areas here, and similarly for auditory right. And I mean, I said scalp areas, I mean, because you cannot interpret this in a straightforward way in terms of brain sources. Unfortunately, sometimes it's done in the literature, but you can already see here that even for an auditory response, which will come from somewhere of the auditory, in the auditory cortex down here, the peak is actually somewhere in, well, you could call it a visual area here in the back of the brain. So the dipole produces a positivity here, smaller negativity there. So you might just mistake that as a visual response. I mean, in this case, we know what happened, so we won't fall for that. But it just illustrates that looking at the data here across the scalp or at the surface or in sensor space can be misleading unless you really know what you are doing. So that's why ideally, of course, we want to find ways of estimating what's going on inside the brain and do source estimation. And that is one part of the following lectures that's going to deal with that. Okay, I think that's the beginning. So the actual data analysis pipeline in MNE Python. So I really just took the overview tutorial from MNE Python. So I do not take credit for this particular Jupyter notebook, but I think it is really a great way to start if you're new to EEG, MEG analysis and you want to know what the software package can do or what you can actually do with the data. So I think these tutorials in general are really a great resource for everyone, and they are openly available. So please feel free to browse them yourself. If you're not familiar with Python, it doesn't matter. I hope it will still make sense. So in Python, you first of all have to load some of the modules or the modules that you're going to use for your analysis. And of course, in this case, also the MNE module. You have to load your data. So define where your data are, and then you basically just say read the data. In this case, FIF is a particular file format for MEG data, might be different in your case. You can then plot some of the, or print some of the information that is that specifies the data. It tells you, for example, that the data have been high pass filtered, 0.1 hertz, low pass filtered at 40 hertz, the number of channels. So there are 306 MEG channels and 70 EEG channels, and so on and so forth. And then you can already plot these data. So you can specify some parameters there. It's not really that important. So here you have a plot of a subselection of channels, and down here, just by moving your mouse, you can just scroll through the data. If I do that, sometimes it looks like, well, there's something going on there. These are probably small drifts, possibly due to movement or breathing, who knows. Occasionally, I'm just looking for something that could be an eye blink here. Okay, some channels have actually, a channel there has actually been, sorry, I can't do that. If it's in gray there on the left, it has been marked as bad already. And occasionally, yeah, you see some deflections there and some of the signals that could be eye blinks. So, well, who knows? Okay, so this is just to show that you can browse through your raw data. You can also then plot the power spectral density here. So this shows you the signal power in decibels, and it clearly also shows that above 40 hertz there isn't really much going on because we had a low pass filter of 40 hertz. So this makes sense. You also get these data for EEG, gradiometers, magnetometers separately. So with MEG, there's the slight complication that there are different types of MEG channels. Gradiometers compute the gradient between two magnetometers close to each other. In this case, they are planar gradiometers, but more about that in a separate section. And magnetometers in particular are more sensitive to noise from distant sources. So from sources that are not inside the brain, possibly outside the magnetically shielded room. And that probably explains these peaks here around 30 hertz, unless these are potentially the head position indicator coils here. Anyway, so you see these peaks, some of these peaks also in the gradiometer data, just smaller. So MEG is more sensitive to external noise sources. That's what we see here. But apart from that, that's how it's supposed to look like. So on a decibel scale, it looks relatively flat. You have a peak here, which I'm assuming in all the sensor types, this is the alpha rhythm for this participant. So this makes sense. And then pre-processing, you will have to do some sort of artifact removal. When I say you have to, you don't necessarily have to, unless if you think that your data are very clean, or artifacts like eye blinks are not systematic with stimulus presentation, then you don't necessarily have to do it. But a very common way to remove artifacts that come from eye blinks or eye movements and the heart is ICA. And this is a very quick way of implementing this for demonstration purposes. So you basically fit your ICA model to your data. You decompose your data into ICA components. In this case, somebody has already looked at the data and decided that two components are bad. I mean, eye blinks and heartbeat produce very strong artifacts. So they say basically that, they explain that elsewhere, and we will talk about that in a separate session. And therefore, I will not explain the details in much detail here. But you can compute these ICA components. You can then look at them. So ICA component 1, for example, has this symmetric frontal topography here for magnetometers, outgoing magnetic fields here, ingoing magnetic fields there. So this is very typical for an eye blink. And ICA component 2, also for magnetometers, has maximum values here around the rim of the sensor array. So there's the MEG sensor array. You're looking at it from the top, left, right, back, and front. And this is quite typical for the artifact of a heartbeat. So they picked these two components already. You can inspect them in more detail, but more importantly, once you've decided what is bad, you can mark them as bad in your ICA model, and then you can say, apply my ICA to the raw data, which removes the contributions of these topographies from your data, and you get cleaner data at the end. So again, I'm not going to explain this in much detail here because we are going to do that in a separate section. So this was just about cleaning the raw data. So whether there are stimuli or not, so you could do it for resting state activity or for anything else. But if you have an event-related design like we do with our visual and auditory stimuli, first of all, we may want to check that these events are actually in our data and that they make sense. So you can detect the trigger values as events in your raw data. So the trigger values will be stored in a particular channel in the data. In this case, it's STI 014. You can check how many are there. Okay, 319. I'm just assuming that's correct. Here they explain what these stimuli mean, but importantly, you can then label these triggers in a more meaningful way. You can basically define 1 is auditory left, 2 is auditory right, and so on and so forth. Then there was the smiley, that's number 5, and the button press 32, but we will not really look at that later. So in the following, in your scripts, I mean, you can really make sense, basically. So you can use the actual labels of your stimuli rather than the numbers. You can plot the events. So this is time. So the experiment must have lasted about 4 minutes, a bit more than 200 seconds. And here you see when these stimuli were presented. It's just sort of a sanity check, whether it's what you expect. For example, if you presented your stimuli in blocks, they should appear in blocks here. You get an idea of how many stimuli actually made it into your recording. You can see also when buttons were pressed, and if the participant pressed buttons at all. And once you think that's all okay, then you can epoch your data. That means you can cut out the data segments around each individual stimulus. But you don't want to do that for epochs that are contaminated by artifacts. So you want to make sure that you at least remove the most terrible artifacts. And for that, we usually define these rejection thresholds. That means every signal within an epoch that exceeds a maximum minimum difference of this value, for example, for magnetometers, gradiometers, EEG, and EOG, will not be taken into account, will not be used as a good epoch. These values may differ depending on your scanner and the purpose of your analysis, and sometimes you may adjust them for different participants. More about that in a different session, but it's a bit of a dark art. It's really only there to make sure you remove the most terrible artifacts, and everything else you should deal with, hopefully with other methods of filtering, and for example, ICA. Also be aware that MNIPython uses the international standard units. So for example, Tesla. So you have to specify this with this exponents here, -15 to go down to femtoteslas, or in the case of EEG and EOG, this is in volts. So -6 means this is in microvolts. And similarly, in other cases, I mean, you specify values in seconds and not milliseconds. So that has led to many confusions already in the past. But if you have all of this correct and you decide what you want to do, then you can epoch your data, the raw data, you specify the events. You specify the events that you're interested in. You need to specify the length of your epoch from 200 milliseconds before to 500 milliseconds after. Stimulus presentation, again, this is in seconds here. Your rejection criteria, and you are saying that you actually want to load the data straight away. It'll tell you which epochs it drops, or at least how many and why. So which channel was responsible for dropping these epochs. It says EOG here quite a few times, which probably means there were eye blinks in there. So in this case, I'm not actually sure whether I would have specified the EOG channel here, because we used ICA to remove eye blinks. So you are removing eye blinks, and then you are still rejecting based on the EOG channels, but that is something you'll have to decide for yourself. And then we can start doing something slightly more meaningful. So we can say, which conditions do we actually care about? So we don't care about the smiley and button presses. We want auditory and visual left and right. We can then make sure that we actually have separate objects here for the auditory epochs and the visual epochs, and right, left here. And then we can, as the very start, plot our data for these epochs for separate channels, for example. I mean, this is something that you can do to either explore your data, but you can also do more interesting things with it. So if you do it for an MEG channel, for one single MEG channel, you can see that, well, this is time, and these are the epochs. So every row of this matrix here is an individual epoch for this channel. You can see that across in this display, you can clearly see that something is sticking out here. So these are individual trials, but if you plot them all together, you can see that there actually is something sticking out from the background noise. So relatively flat baseline. And then at 100 milliseconds, this is probably, let me just check, is this the auditory epoch? Yeah, this will be the auditory P1. So it is present in almost every trial. And in the average, you can clearly see this deflection here, 100 milliseconds. And I actually think that's quite nice. So especially if you then have something like reaction times, for example, or other parameters. I mean, if this was a bit more interesting, like a word or a picture, you might have something like word frequency or difficulty or something like that. You can sort these epochs based on some other variable, and then see if also which brain responses actually depend on your variable. So this gives you an opportunity to explore your data. This EEG channel here, it's indicated here where this channel is. It's actually not in a very exciting place. So you don't see a huge response here, but still in the average, at least you see a little deflection here at 100 milliseconds. Okay, so you could also decide now to look at your data in the time frequency domain. We will talk about this later, but for that, you just have to specify a frequency range, in this case from 7 Hertz to 30 Hertz in steps of 3 Hertz. And this is all done I mean, just for demonstration purposes. So you might want to specify a larger frequency range or a smaller step size here, but this will take longer. So don't think that everything here in these demos is necessarily the way you have to do it. You have to specify something that's called the number of cycles. That basically determines a little bit your frequency, time and frequency resolution. More about that in another session. But if you do that, you can plot your data in this frequency time domain for one channel. In this case, we are just looking at an auditory evoked response, which means this response has a frequency content just below 10 Hertz. So it peaks at about 100 milliseconds here. So we don't necessarily see anything new with this display, but it just shows you how quickly you can get this if you want it. More common is the estimation of evoked responses. And for that, you simply take your epochs and you say average. And then you can visualize them. In this case, you can plot them both together, or you can plot them separately. So this plots the global field power. So it computes the power across all the senses. It's not surprising that auditory and visual responses differ here, but if you have a more interesting experiment, you can of course check whether there is any difference across senses at all between your conditions, for example. But this at least looks like a clean evoked response. So for a sanity check, I think that's quite nice. So you have a baseline which is similar between your condition, and then you have the kind of evoked responses here, the kind of time course that you would expect. Okay, you can look at that for different channel types. So magnetometers, gradiometers, so these are the two MEG channel types, and EEG. So you shouldn't plot all your channels in one display because they're in different physical units. But sometimes it can also be interesting actually to track your channel type. So whether an effect or a response is only present in one or a subset of your channel types or in all of them. And then there are different ways of visualizing your evoked responses. You can plot time courses for all your channels plus some topographies in the same plot. You can only plot the topographies. So this would be EEG topographies here. This would be the magnetometer topographies for your auditory response. You can compute different waves and then plot them for all the sensors. you have to decide for yourself whether that makes sense. I mean, often it's hard to interpret this, but you may just want to check if there is which sensor groups might show the biggest effect, or it's also just maybe to see if there are still some noisy channels in there. But once you're convinced that you have a clean evoked response, then you can ask yourself, okay, where are the generators of this response? So can we actually find out where in the brain these signals come from? And that is called inverse modeling. And for source estimation, you need an inverse operator, which in this case has been created before, as many other things. We will talk about that later. But there's a relatively standardized pipeline to do that. So you read your inverse operator. You need to specify a signal to noise ratio. Python assumes or suggests usually a signal to noise ratio of 3. There is no really precise answer to choice for this parameter, because the signal to noise ratio will change from the baseline across the time course of your data. So you just can find a compromise or something that's approximately right. And in later sessions, we will see that it is actually your analysis is usually quite robust with respect to this parameter. So, and with this, you are in business. So you can say, apply my inverse to my data, in this case, the visually evoked responses. Use your inverse operator, specify my regularization parameter, choose a method. There are a few available. Just to show that because things have already been prepared here, this actually happens quite quickly. So it's basically matrix multiplication here. And once you've done that, you can visualize your result. And I'm just rescaling the data a bit for visualization purposes here. So this is what we are after. So here we see the left hemisphere, the right hemisphere. It was the stimulus, the checkerboard presented to the left visual hemifield. So we clearly have a right lateralized response. We are at 100 milliseconds after stimulus onset. So that makes sense. So we can potentially move around here and also rescale the display. Whether that makes sense or not is a different question. So let's maybe go back to roughly the 100 milliseconds here. But it doesn't matter that much. Let's rescale. And well, as so often, scaling plays, of course, a big role here in the possibly also for the interpretation of results. So I usually prefer a more honest scaling that starts close to 0 and maybe has scaling stuff, the color scaling that's continuous between 0 and maximum. But if we do that, we can clearly see that we have a nice focal response here in the visual cortex. Spatial resolution and localization accuracy, of course, is a big topic for EEG and MEG source estimation. The visually evoked response is probably not that widespread. It's probably more focal here in visual cortex, but with EEG and MEG, you have limited spatial resolution. You get a blurred version of the true underlying cortical sources. That is a big topic that we will deal with in a separate session. So there are ways, at least, to deal, or at least to describe the spatial resolution of your source estimation methods. But this is at least the kind of resolution you can expect, at least in a best case scenario, because here we can use combined EEG and MEG. We have relatively high signal to noise ratio because we have quite a few trials in here. And also we are looking at the brain region that is well covered by the sensor array. So the sources are quite close to the sensors. So do not expect spatial resolution to be that good across the whole brain, and especially not for deeper brain areas. Again, this is something that we will talk about in a separate session. But for now, this was the overview, at least of the analysis pipeline in Python that leads from the raw data over evoked responses to the source estimates. So this is something that can already be the basis for many, many papers that you see in the literature.